relates to, let me go answer your question by way of going and I'll come back to your question. What you need in order to have a good microscope, you have to have things magnified. But just to increase the magnification of something, if it's blurry, it is useless. And so the other need is to have high resolution. In other words, to see it bigger, but also to see it more clearly. And then, of course, the third criteria you need is if you were to just take a cell out of the body and look at it in a microscope, you would see nothing. And so you need special dyes called stains. Coming back to your question, what the oil is doing, when air is going through a lens, the air part, air is molecules. And as the air goes through the light, when the light is shining through the light source, the light is scattering the air particles, and that scatter makes things look blurry. And so what the oil is doing is it's a more viscous solution, and so when the light comes through, it concentrates the light to a point. If you've ever looked at your windshields, and if they're dirty and you're driving into the sun, you have dust particles that are on the windshield, and you can't see anything. You see all these rainbow of colors. And so what the oil is doing is concentrating everything to a point so you're seeing clearly. That's why if you try to use an oil immersion objective without oil, you're right back to a problem that's worse than when it started. Everything was blurry. So the oil is meant to be used with the light microscope on only the 100. So that's the only glass that has been ground in a certain shape that works with oil. So if it says it is an oil immersion objective, you have to use it with oil. And so the idea would be it's going to increase your resolution. If you're taking microbiology, where you're going to be spending a whole term, I don't know if people in LAX will be, I think that's part of the curriculum. There you're spending a whole semester looking at bacteria, which are even tinier than human cells. You would not be able to see anything if you didn't use an oil immersion lens. So that's where you're really going to get most of the experience with oil immersion. So here's where we relate to our lab. There is, in theory, if you were a science fiction writer, you could possibly imagine building an organism that's very large that also is composed of just a few huge cells. But the reality of life is that most organisms, we're fairly large in the scheme of things, but our cells are only maybe 3 to 5, maybe maximum up to 100 micrometers. And so the only thing that separates most people from scientists are very curious, and so the question arose very early, why is it that you have a limit to cell size? And that was the whole purpose of the lab. And so the limit to cell size is because of the requirements that the cell needs. The bigger the cell, the more the volume. And those of you that did the lab have already done all those calculations. So we have some reality. Volume is, here's a big cell, here's a little cell. This obviously has more volume. Volume determines the need that the cell has for nutrients. But the only way that the cell can take in nutrients and get rid of waste products is through the cell membrane. And the cell membrane, of course, is around the perimeter. So that in the cell membrane, in the perimeter, the circumference of the cell is determining the ability to meet the need. And so the cell membrane would be related to the surface area. So surface area, volume determines the need of the cell. The bigger the volume, the more the need. And the surface area with the cell membrane around the circumference determines the ability to meet the need. And as the focus of the lab is, just by mathematics, if you start trying to do the calculations of a diameter of a cell and the surface area, the volume of the cell and the surface area of the cell, you find that you run into a problem. And the problem is indicated by the surface area volume dilemma. And the dilemma is that if the cell is too big, the surface area is not going to be able to catch up. It's not going to be up to the job of allowing things to move in and out of the cell according to need. You don't have to write this down because you have it in your lab manual, so just concentrate on here. Here they're showing you the radius of the cell. A small cell would be 1 unit. A large cell is 10 units. If you do the calculation, you find that the surface area here is 12.57 units, and this is 100 times more. And so you have a problem that there has to be some limit or some adaptation that a cell has to allow the surface area to catch up to the volume. And so that is the basis of the whole lab that we're doing. The lab is essentially showing you that if we just consider diffusion as a way of getting things in and out of cells, things moving down the gradient from high concentration to low, if the volume of the cell is too big, the diffusion rate is so slow, the cell would die before anything else happened. And so the idea is in your lab manual, you have a whole list of adaptations that the cell has to develop. Either the cell stays small and solves the problem that way, or it's going to have a lot of adaptations that we'll discuss in a minute. So here is the problem. Well, this is the solution here. These dogs are different sizes, but their cell size has remained fairly small. If you were going to take a cell out of a small dog and measure its diameter, and take a cell out of a large dog and measure its diameter, it's going to be pretty much the same. So that's the solution that we have adopted. One of the solutions, I should say. We just have small cells. We didn't develop into a 6-foot individual with a commensurate huge cell. And so here is one solution to that dilemma, what is called the surface area to volume dilemma. One is to avoid the problem. So if you stay small, then you don't have that surface area to volume dilemma. And so here's exactly, this is the one from your textbook, I think, and this is showing that problem here. Here's a small square. Here's your surface area to volume ratio. Here's a large square. If we had cells that got larger, notice what happens to our surface area to volume ratio. We've gone down almost 6 times. That would not be enough to allow the cell to survive. So what's our solution? We have the same size overall, but we have a body that's composed of many small cells. And so we have avoided the surface area to volume ratio problem if the cells in our body stay small. Yep.
If you have like a larger, if a tall person has like the same exact cells as a regular average tall person, but they have to keep on dividing in order to work and maintain that height and maintain that growth.
Well, we're talking about the problem of one cell. So in that respect, we can get to be 6 feet tall, 7 feet tall perhaps, and still not have this problem, because no matter how tall you are, you're still going to have an individual cell will be more or less the same size. And so this is the solution that all bacteria have adopted. Bacteria, almost by definition, are unicellular. We are multicellular. And so that is one of the defining characteristics of an animal. The organism is composed of many cells. Bacteria have solved their problem forever by just remaining a unit. They've never developed a colony to become an organism that we think of. So they are defined as unicellular prokaryotes. And we'll talk about other things that are prokaryotes, but right now the most familiar ones are bacteria. So that's their solution. Just remain small and avoid the problem completely. You can find a geometric solution. So you can get a little bigger if you change the shape of the cell. Notice that we were talking originally about a sphere, like actually in diameter be a little bigger than normal, but they're going to be able to have diffusion because they're so thin. And so we have some cells in our body that look like pancakes, very flat, but a little longer. Some bacteria have solved that. There's a bacteria known as bacillus. We don't have to dwell on it. You don't have to know the name. But a bacillus is a rod. We will actually talk about those a little bit when we get into the prokaryote chapter. And that bacteria is a little bigger than most bacteria, but it's okay because it's flat. It has flattened out on the width side. The cells that are lining your cheek are also flattened out. And so the cells that line your blood vessels have flattened out like pancakes. And so that allows, because there's not much cytoplasm or cytosol inside in the width division, that means blood can move back and forth across the cells very easily. So that's another solution. Here's a third solution. You can increase your surface area. That's a very common solution. If you go into the lab and you look at the torsos that are in lab, you see our whole intestine is all coiled. If you look at the brain that's in the cranial cavity, you can see the brain is all folded. So folding is a very common solution to increase surface area. And so here is an example of cells in the intestine whose membranes are actually folded. So you have folding upon folding upon folding. You have the intestine with some cells that are folded, and then if you look at the individual cells that are lining the intestine, those cells are folded. And so the folding increases surface area. And so that allows nutrition to be able to move back and forth across the body. You can eat food, and you can make sure that it gets out into the bloodstream to serve the cells of the body. This is the roots of a plant. The roots have rootlets. So the main root is here, and then out of the root are these little hairs, we call them root hairs. All those root hairs are doing is increasing surface area. So the plant, the only way a plant can get water into its internal being is through the roots, and so having root hairs increases surface area. So even plants that are living in areas that don't get a lot of rain, any little raindrops that come along, they're going to be able to absorb the water. Here is another strategy. This is one that's used by plants. Have a big cell, but don't have all that cytosol. Don't have the volume that has to have nutrients diffusing back and forth. Carve out the middle of the cell with a huge vacuole that just holds water. And then the water, and so now you have two things going on. You have eliminated the need for you have eliminated the volume, and you also have provided a little storehouse for where water that's picked up by the root can be stored to the stem of the plant. And so you've done double duty in that kind of adaptation. So, and in carving out that central space for the vacuole where the water is, you don't have as much volume. So this is a very common strategy that most, if not all, plants have developed. Another strategy is just get to be a little bit more of a complicated organism, but develop adaptations that allow you to increase your nutrient supply. So that's another way. And so one adaptation is develop muscles. That was one of the very early adaptations in evolution, where a muscular system was developed with muscles that were able to contract and move the organism. And so now you can move to where the nutrients are. So even if you have a large cell, you're able to constantly feed it and not allow it to dry out or run out of its nutrients. Protists are a certain kind of cell that we'll talk about. We have a whole chapter devoted to protists. Most of them live in the water. Most of them have means by which they can swim through the water. A lot of them are attracted to light, so they can go towards algae and things that are nutritious for them because they have sensors that attract them over to that area. And so you really can't, can't increase your nutrient supply unless you have developed a body structure that allows movement. And that's why the basis for a lot of the early evolution in living things was a muscular system. To either swim, and then eventually when the animals moved on land, to be able to develop limbs that allow you to move from place to place. Some bacteria have that same movement system. They have a tail called the flagellum, and that allows them to swim through fluids to be able to move wherever food supply is. Some of them have sensors that they can sense where chemicals are that are in abundance. Another solution is to be able to actually be a hog, in a way, and just gobble up huge amounts of food whenever you do find it. So you don't have to constantly be moving to where the food is, but you can take in such big amounts that you satisfy the nutrition demand. And if you know what amoeba are, they're phagocytes. Phagocytosis is the ability to extend the membrane, engulf something, and then bring it into the body of the cell. And so amoebas have that ability, so they can increase the body size by about they're a single cell, but they can increase their size by 2 and 3-fold after they've ingested. Usually what they eat are other protists or bacteria, and so they can get a huge meal all at one time just by ingesting it. So these are all amoeba, paramecium. These are all single-cell organisms. Here is another strategy, is to just get more efficient in what you do. And so when your plant cells and amoeba are also capable of what's called cytoplasmic screening. When they ingest something, they have a mechanism by which to digest it with enzymes and then have it move rapidly from one place to the other in the cell. And of course, we, in a sense, as a human organism, have that ability too. We have a circulatory system. So every time we eat a meal, we can digest it down to the monomers, and then we have a bloodstream that sends it off to every part, every cell in the body. So all of these have been adaptations that are coming from that essential dilemma of cell surface area and cell size. So we have developed, and volume rather, of a cell. So we, as humans, have a much more sophisticated resolution to the problem than bacteria does. The bacteria, most bacteria's resolution simply just stays small. But we develop muscles, we developed a nervous system that can control the muscles, so we can move in a directed manner, etc. Plants have developed a fairly sophisticated solution by having vacuoles that also serves to distribute water, which allowed them to get taller. They didn't have to remain short. So these are all things that all of these problems and all of these adaptations are coming from that essential problem relating to that surface area volume, and that's exactly why we have a whole lab devoted to it. It's kind of the spur, the need that created the evolutionary adaptations. Okay, so here are some other, now we're heading towards even more sophistication in terms of our cells. Improve the efficiency of what happens when the nutrients get into the cell. And so one way is to have a division of labor. You know that you can get more work done if you have a lot of people helping than if you try to do it all yourself. And so one of the things we'll be talking about in the cell is the idea of compartmentalization. A eukaryotic cell, we are a eukaryote. We have a lot of membranes inside our cell. And the membranes that are folded up inside our cell are increasing surface area and allowing different events, chemical events, to take place inside the cell that ordinarily wouldn't happen if we just had one nucleus and the rest being cytosol. And so this is the solution that we have adapted. Not only do we have all the other solutions that we just talked about, but we in addition have adaptations within one cell itself, and that is that compartmentalization. And I'll give you a few minutes to write it down. The other solution we've adopted is we've gone away from the bacteria strategy of staying small and just being unicellular. We are multicellular, so that's another kind of division of labor. We have, at the very beginning of the term, we talked about the organization, the system, the organism being composed of systems, the systems being composed of organs, and so all of those systems are divisions of labor. We have a respiratory system to get oxygen into our body. We have a digestive system to get glucose into our body. We have a urinary system to get rid of the waste. And so that becomes another kind of efficiency that only a multicellular organism would be able to adapt. So with each of those increases in sophistication of adaptations, we are a lot more able to do more things than a single little bacteria sitting in a dish or a body or a pool somewhere. So most of the animals or organisms on Earth have adapted this multicellular strategy. Animals, plants, most of the fungi and algae. Algae are little, usually photosynthetic green things that people are what people call pond stuff, that little green stuff that you see. It's like little hairs on the ocean. That's little algae. And so they're actually living things that we'll talk about when we get into the protists. All of those have adapted some level of multicellularity. So it gives them a lot of a big range of activities that they can be in. If you had, here's a little mouse. Now he is small. His cells are all the same size, but we do obviously on this sort of have a different range of sizes of animals. And so even though each animal is has more or less the same size cell, the very fact that we have a small mouse and then we have a large elephant, they still have to deal with a surface area to volume problem. And so this little guy has a high surface area to volume ratio. And since that is the case, one of the things that's going to happen, we talked about metabolism as being a chemical reaction, that and every chemical reaction generates heat. One of the problems these little animals have is thermoregulation. They lose heat very rapidly. And so what their adaptation gets some fur. If you have fur on, then the heat is trapped in your body. You don't lose it as rapidly. One thing we know, if you're cold, you kind of shiver and you wrap yourself up in a ball. My dog is cold and he's outside. He's all curled up in a ball. All animals do that. That turning into a sphere is keeping the heat in your body. So this is a behavioral adaptation to the problem of losing heat because of because he is small, he has little volume. So relatively speaking, he has more surface area. So this is the solutions of animals that are in fact small. You have large animals that live in cold weather, and so now they have the opposite. They have a low surface area to volume ratio. That problem, when you have a low surface area, you have the potential problem of not being able to effectively generate heat, where you just can't get up the metabolic rate. And so the adaptation is live in a cold climate where you don't need that much heat, where you don't need to, you can have a low metabolic rate. We know that if you go into cold climates, our metabolic rate decreases. And so this is a solution that these guys have. If you're large and you live in a hot climate, now you have to be able to dissipate heat. We used the term evaporative cooling before. And so this is the solution for an animal that lives in the tropics like an elephant. Don't have fur, so that the heat can be dissipated very easily. Have wrinkled skin. All of these wrinkles on the elephant's hide is increasing surface area. And so if you increase surface area, you're going to have a greater surface area to allow the heat to escape. And have large ears. We started our discussion of homeostasis with that, the jackrabbit with the blood vessels that allow heat to escape. So these are the solutions to that surface area problem when they are in a climate that is hot and they must lose heat, otherwise they're going to die of too high of a core body temperature. Okay, so now we're moving away from the problem of a cell and the problem of animals that are multicellular and how they solve their problems of being having that surface area, the volume dilemma. And we're back to a review of the different kinds of cells that exist on the planet. These are the three domains. We had this question at the very beginning of the chapter of the semester. So we're back again to life is divided into three domains. We have the bacteria and the archaea, which are the domain of the prokaryotes. Karya means the nucleus. So prokaryote is before the nucleus developed. Archaea, remember, are the extremophiles. They're the ones that live in those very hostile environments of the hydrothermal vents that are up to a few hundred degrees in the Arctic and cold. It's high salt water, the Dead Sea, which is about 10% sodium chloride. They can survive in those areas. And then we have the eukaryotes, so fungus, plants, animals, and things we probably haven't heard of that we'll talk about as we get into the chapters. All of these are our domain. We should be well versed in that. We've had a quiz and an exam on this before. And so here is here's one example of a prokaryote. Here's one example of a eukaryote. And there are some things that are different, but there are other characteristics that are common to all cells in all domains, regardless of whether they're prokaryote or eukaryote. One, everybody has a cell membrane or a plasma membrane. Everybody has that selectively permeable membrane to allow things to move back and forth. Everybody has cytosol, that fluid-like substance that is surrounding the organelles that are in the cell. Everybody has DNA. I think that was a question on either a quiz or an exam. What's the commonality? Everybody has chromosomal DNA, which means everybody has genes. And so that too, the genetic inheritance of all living things on Earth. And everybody has ribosomes. Ribosomes, as we'll see, have the function of making proteins. And one important chemical that's a protein are enzymes. And we already know that enzymes are catalysts. They increase the rate of reaction, and every organism undergoes chemical reactions. So we're not too surprised that they have ribosomes. So these 1, 2, 3, 4 things are common to all living things. And then we have some things that are different. Prokaryotes, which are bacteria, have a cell wall in addition to a cell membrane. And so the cell membrane would be here, and the outside with all these little extensions that look like cilia would be the cell wall. It's the cell wall that if you have an infection and you've gotten sick because of some bacterial infection, most of the antibiotics are dedicated to acting on the cell wall. They're going to attach to the cell wall. So cell walls are unique to the prokaryotes as opposed to, well, I shouldn't say that. Plants have cell walls too, but let's leave it for a moment. A nucleoid region. Notice that the eukaryote has a membrane-bound nucleus. If you look at where the DNA is sitting inside the membrane, sitting inside the nuclear membrane or the nuclear envelope. If you look here, the DNA of a prokaryote is kind of all just kind of floating around in the cytosol. It's not bound by a membrane, and so it's called a nucleoid region. And so this is a huge difference. No they're still dangerous because they can invade our body and start doing their metabolism, producing chemicals that make us sick. But in terms of evolution and simplicity, they're much simpler than multicellular organisms. So these are ways they're different. Here's our eukaryotic group again. We have whole chapters coming up on plants, on animals, on fungi, on protists, those little algae, etc. Notice that one of the defining features of the eukaryotic cell is that it is multicellular, as opposed to the prokaryote, which by and large is unicellular. Okay, so here is our eukaryotic cell, and here is our compartmentalization. Here's our nucleus, surrounded by a nuclear envelope, as it's called, which is the membrane. And then extending from the envelope of the nucleus are other structures that are enclosed by membranes. The mitochondria enclosed by the membrane. And so this is what we mean by compartmentalization. This has a function, this has a function, this has a function, this has a function. So you now have compartmentalized organelles. An organelle is a little organ that's sitting inside a cell. Each of these things have different functions, which is the whole subject matter of the chapter. What are those functions? Here's a plant cell. Plant cells are also eukaryotes. It has membrane-enclosed organelles. It has, in addition, something we don't have. This has that vacuole that we were just talking about, that central area that's carved out just for no other purpose than carrying water. And that vacuole is partly what makes plants able to get so tall, because they can, if you're 60 feet tall, you can be sure that the water is going to move up by capillary action because of the surface tension and all the properties of covalent water, and it's going to move right up from the root, up through that central vacuole into the leaves, and so it can remain alive. So here's the organelles that we want to know. We want to know, I'm just going to go over them, and then next time we'll talk about them in detail. We want to know, number one, the nucleus. And you want to make sure you know that the nucleus is bound by a nuclear envelope with pores. We'll talk about what the pores are. Things actually have to move in and out of that nucleus. And they're moving in and out through the holes or the pores in that nuclear envelope. Number two, we want to know the ribosomes and what the function of that is. We just talked a little bit about it. Ribosomes are where the proteins in the cell are made, which include enzymes. Number three, we're going to talk a little bit about these two organelles, the endoplasmic reticulum and the Golgi apparatus. And let me add to that 1, 2, 3, 4. And number 5, we're going to talk about the lysosomes. And what they're doing in the plant. So we're going to concentrate on some of the major organelles. And of course, how can I forget, number 6. Number 6 is the mitochondria. So those are the major organelles that we'll talk about in terms of their function next time. Okay, so let's call it